Hello and welcome everyone. In this video, I am going to discuss a very important topic, cyclic and non-cyclic photophosphorylation. This is also a very important portion for NEET biology and students need to have a clear concept about this portion to answer the questions from here. This portion carries a weightage of 13%. So, let us follow this slide. The first slide is of cyclic electron transport chain or cyclic photophosphorylation. So let me give you a brief idea about what is cyclic photophosphorylation. First, let us look at the diagram. So this is the cycle or the diagram of cyclic photophosphorylation. When the molecules in PS1 is excited, electrons are released. Okay, so photons are falling in this antenna complex. Electrons are getting excited and released. The electrons are captured by ferredoxin. Through ferredoxin reducing substrate. There is a substrate named ferredoxin reducing substrate. Electrons are captured by this ferredoxin with the help of ferredoxin reducing substrate. Due to the non-availability of NADP+, electrons from the ferredoxin moves to photosystem 1. So from this ferredoxin, due to the non-availability of NADP+, electrons directly move to the photosystem 1. So because of the direct moving of electrons from ferredoxin to photosystem 1, this whole system is called cyclic photophosphorylation. So these are the electron carriers. Okay, these electron carriers helps in transferring electrons to the photosystem 1 again. So electrons from ferredoxin moves through photosystem 1 through electron carriers like ferredoxin, plastoquinone, cytochrome B6F, plastocyanin, etc. Okay, so here two molecules of ATP is released. Okay, no NADP plus is released, only two molecules of ATP is released. So let us go through the points which are included in this slide. First is in cyclic photophosphorylation, PS1 works, only PS1 works. Okay, cyclic ETS or PS1 is activated by wavelength of light greater than 680 nanometer. Very important point, students. Cyclic ETS or PS1 is activated by wavelength of light greater than 680 nanometer. It occurs in the grana thylakoids and stroma thylakoid. These are the locations of cyclic photophosphorylation where cyclic ETC occurs. During cyclic ETS, the electron ejected from the reaction center of PS1 returns back to reaction center as I showed previously in the diagram. In cyclic ETS, no oxygen evolution occurs because photolysis of water is absent. This point is also very important. Actually, all the points which are included in this slide is very important. The photophosphorylation takes place at two places, thus 2 ATP generate at each ETS. Okay, very important, 2 ATP generates at each ETS because photophosphorylation takes place at two places. One at the grana thylakoid and other at the stroma thylakoids. Okay, so NADPH2 is not formed in the cyclic process. As I previously also mentioned, no NADPH2 is formed, only ATP is formed. Plastocyanin is Cu containing blue protein in cyclic ETS. This is also a very important point. Plastocyanin, it is a Cu containing blue protein. Okay, like copper containing blue protein. Next we move on to non-cyclic photophosphorylation. Okay, so let me give a brief idea again what non-cyclic photophosphorylation is all about. When molecules in the PS1 is excited, electrons are released. So here electrons are excited when light falls on the reaction center. The electrons are now transferred to NADP+. Okay, electrons are now transferred to NADP+ with the help of the reaction carrier molecules like ferredoxine. Okay. When the molecules in the photosystem 2 is excited, here also when light energy falls on this reaction center, electrons gets excited. The electrons are transferred to photosystem 1. Okay. So when electrons move from photosystem 1, the electrons in the PS1 gets reduced. To compensate the reduction of these electrons from photosystem 1, electrons from photosystem 2 moves to photosystem 1 with the help of what? With the help of this electron carriers like pheophytine, plastoquinone, cytochrome B6F, plastocyanin, etc. Now, since this process occurs in Z manner, as you can see here, it is forming a pattern of Z. So it is called Z scheme. So what? So non-cyclic electron transport chain or non-cyclic photophosphorylation is only called Z scheme because it is maintaining a Z-like pattern. Okay. So let us follow this slide. It has been written here. Both PS1 and PS2 is involved in non-cyclic electron. In cyclic electron only what? Only PS1 works. Only PS1 is involved. But in non-cyclic photophosphorylation, both PS1 and PS2 is involved. It occurs at grana thylakoids only. Okay. In cyclic photophosphorylation, two locations are there, grana thylakoids and stroma thylakoids. But in non-cyclic, only grana thylakoids are involved. The location is only grana thylakoids. Now, the electrons ejected from photosystem 2 never go back to chlorophyll A680 and finally gained by NADP. During this process, oxygen is evolved due to photolysis of water. Very careful, in cyclic photophosphorylation, photolysis of water is absent. But in non-cyclic photophosphorylation, photolysis occurs and formation of NADPH2 and ATP occurs. Okay. So previously what we saw that in cyclic photophosphorylation, no NADPH2 formation occurs. But in non-cyclic photophosphorylation, NADPH2 and ATP both occurs. So you need to remember all these points very carefully before the exam, and these points are very important and you are definitely going to get questions from all these points. Okay. And all these points mentioned in this slide will give you a clear concept about cyclic and photophosphorylation, cyclic and non-cyclic photophosphorylation. So before exam, it is must that you go through this slide. Okay. Now comes to the difference of cyclic photophosphorylation and non-cyclic photophosphorylation. The first point is it involves photosystem 1 and non-cyclic photophosphorylation involves both PS1 and PS2. Okay. Second point, it is not connected with photolysis of water, so no oxygen is evolved. In non-cyclic, it is connected with photolysis of water and liberation of oxygen. Third point, electrons moved in a closed circle. Electron expelled from P700 returns to it after passing through different electron acceptors. I showed this thing in the diagram also. Just followed by previous, rewind it and you will get that I have told that in cyclic photophosphorylation, electrons come back to photosystem 1. Okay. Electrons do not move in closed circle. Electron expelled by the reaction center P680 does not return it. Here water donate electrons to P680. Okay. In the fourth point, in each flow of electrons, two molecules of ATP, very important students and NEET aspirants, Two molecules of ATP are synthesized in cyclic photophosphorylation. But in non-cyclic photophosphorylation, only one molecule of ATP is synthesized. You need to remember this thing very carefully because this is very important and tricky part, and you need to remember this, okay? Now, NADPH is not produced. I told previously also NADPH is not produced in cyclic photophosphorylation, but in non-cyclic photophosphorylation NADPH is produced, okay? Now DCMU. DCMU is an inhibitor of electron transport chain, okay? So cyclic photophosphorylation is not inhibited by this compound DCMU, but in non-cyclic photophosphorylation or in non-cyclic photophosphorylation is inhibited by DCMU. Okay, I hope all this you will go through all these points before examination because these are very important, and this slide itself is very important and very informative. Before exams, students, please go through this slide, okay? Now I am going to discuss few questions which has been asked in the previous years in NEET exam. You will get more questions from the net also or from many books which are available in the market. But I am going to discuss these two questions for you. Okay. So the first question is, cyclic and non-cyclic flow of electron is used in plants to A. Meet the ATP demands of Kelvin cycle. B. Avoid producing excess NADP plus H plus. Balance ATP and NADPH plus H plus ratio in the chloroplast. All of the above. So the answer will be all of the above. Okay. Easy. Now second question. Cyclic photophosphorylation results in the formation of A. ATP and NADPH. B. ATP NADPH-O2, C. ATP, D. NADPH. So very clear, if you go through my slide you can easily answer this question. C. ATP is the correct answer. If you like this video, students, please subscribe our channel and do like, share and comment. Thank you.